Alright, with this video I want to show you a cereal dilution. So to begin with, I began by autoclaving a bunch of tubes. These tubes with the green cap have no water in them at all. These tubes with the white caps have water in them. Now, cereal dilutions, you need an exact amount of water. When you autoclave, you don't know exactly how much water is there because some is lost as steam and some may boil over. Therefore, what I am going to do is I'm going to pipette exactly 9 milliliters of water from each of these tubes over to these 5 tubes. Okay, so I have my sterile pipetters. I'm going to do all of this procedure underneath my flame so that it is sterile. I will grab my 2 tubes. We will flame the tops of my 2 tubes, and then I will just transfer 9 milliliters of water. And I'll continue that until I get 9 milliliters of water. And then I will fill up each of these tubes then with 9 milliliters of water. So I now have 5 tubes. Each of those tubes has 9 milliliters of sterile water ready to go. I have a soil sample. This soil sample came from Boulder Creek right down here, right next to the water off of campus. I want to take 1 gram of this soil sample and put it into my first test tube. So we'll put my weigh boat on. We will tear my weigh boat. Now what I want to do is get 1 gram of soil. Patience is important. There we go. I have exactly 1 gram of soil. Now I want to take my 1 gram of soil and get it into my test tube. Have the 1 gram of soil in the test tube. Now I want to vortex it. I just want to vortex it for about a minute. I've finished vortexing it for a minute. Now I want to get my Bunsen burner going. Alright, and I want to then transfer 1 ml from 1 tube to the next tube. Okay. Alright, now I'm just going to mix it up a little bit right before I transfer. So I'm just going to go up and down to mix that up. And we'll get our final one in here without getting too much debris. And I'm going to transfer then my 1 milliliter into my second tube. Alright, we'll get rid of our pipette tip. We will then transfer 1 ml into my third tube. We'll vortex this for a second. Alright. Flame those 2. I'm going to mix it up again right here with my pipetter. I'll put 1 ml into my third tube. Alright, we'll get rid of our pipette tip. grab another one. We'll grab another tube. Okay, we'll go ahead and vortex that a teeny bit. Alright, flame it again. I'll pipette up and down to make sure that it's mixed up. We'll do one on our next tube. Make sure that we don't mix that up. Then we'll go to our final tube. Get rid of our pipette tip. Grab another one. And then we will get that lids done. We'll vortex this a teeny bit. Okay, I'm going to mix it up some in here. And then make that transfer. Okay, and there we are done. Now I need to make sure I label each of those tubes. 10 to the negative 1, 10 to the negative 2, 10 to the negative 3, 10 to the negative 4, 10 to the negative 5. So now we're ready to go on to the next step. I have here my tubes of soil samples. Here's my 10 to the negative 1, 10 to the negative 2, 10 to the negative 3, 10 to the negative 4, 10 to the negative 5. I have 11 plates here, 5 plates of TET 0. 5 plates of TET 3, 5 plates of TET 30. These are labeled all ready to go. You can see this one is for TET 0, dilution 10 to the negative 1. We'll do that one as an example, then I'll continue with the rest later. First thing I want to do then is I want to take my sample and I want to vortex my sample. Get it all nice and mixed up so that we're all ready to go. My pipetter here is set at 200 microliters. We'll get a pipette tip, pull down the plate, get right side up. We'll go in then and get 200 microliters. My sample. And put it on the plate. Now here we have sterile beads all ready to go. So just want to take my sterile beads here and we'll put a few sterile beads down on my plate. And then we'll just kind of move these back and forth so that I can get nice and good mixture of everything on there. You notice I don't go in a circular pattern. I move back and forth in every which direction so that we get a nice, good, even distribution of everything on there. And we'll dump the sterile beads in there. And then this is all ready to go. We'll incubate that at 28 degrees for 3 days.